Eastern Illinois University symposium about science and technology, or on science and technology. Revolutions in science and technology paradigm. What had this to do with paradigms and revolutions? That's what we will get to know with our esteemed presenter, Dr. Gopal Periyannan. Please join me in welcoming.
Thank you very much for the invitation. Again, we care about two things. The earth should be stable. Katie talked about that. And next one, you care about your body, isn't it? So those are the two things. So here, let's talk about a little bit about the things happening inside our body. So this is a topic here. So the body is about bacteria we're starting with. So we all live with many bacteria. Bacteria comes in different size, shapes, and different features, as you know. One of the features is some bacteria contain this flagellum. So that's the way they move around. So they flip that one so they can go and find food or stay away from toxic materials and those kind of things. So what is this real bacterial flagellum? Of course, it has biological material, but if you dig deep into that one, for technology folks, you'll be interested to see that one. It is actually a molecular motor. Okay, so you see a rotor and a stator. Okay, so both parts necessary to make a motor. So this is actually the architecture of this flagellum inside. But of course, we know that when bacteria do not carry nuts and bolts or metal plates and things like that, so these has to be either proteins, carbohydrates, lipids. These are biomolecules. So in fact, if you look at the great detail of a bacterial flagellum architecture, it's a wonderful machine. It is built with several proteins, actually 43 different proteins coming into making this one bacterial flagellum. And it is just settled in between two membranes. Here we are looking at gram-negative bacteria and gram-positive bacteria. There are two different types of bacteria. And their cell wall is different. Here we are looking at gram-negative bacteria. It has a outer membrane and an inner membrane and a space in between that membrane. And so the 43 protein assembly, the bacterial flagellum sits on it and stably positioned with the peptidoglycan layer, this like a chocolate slab-like structure. It's a protein and carbohydrate. It holds the assembly stably in that place. And so this motion takes place right in this region where the inner membrane. So on this inner membrane bound, this set of protein contains a couple of proteins, and then one of them is called this MOT protein. So it comes in two types, MOT A and MOT B, and that is the portion which is really making the motions so that the bacteria can whip its flagellum so that it can move around. So if, as technology people or physics people know that if it's a motion, show me the energy. Okay, you will always ask for it. So where does the energy come from for this motion? It's again all about concentration gradient. So things happens in biological system depending on the difference in concentration gradients. So here, even within the bacterial setup in the membrane, actually the concentration gradient of proton is building up. So what really happens is between the bacterial membrane, two membranes, bacteria uses some proton pumping proteins. So these proteins are membrane-bound proteins, and they pump protons using different sources of energy. It can be a solar energy. They use the solar energy and pump proton into the inter-membrane space, or they can pump proton from inside cell into the space using chemical energy. So using this, they pump the protons into this intermembrane space, so they are concentrating the proton into that intermembrane space. It is simple analogy like you pump your water to a high water tank where you have high potential energy water which flows when you open the tap. Okay, so very principle wise that is the same thing. But here you can appreciate that one. What essentially happening is this energy, for example, solar energy is actually converted into chemical energy which is stored in between this membranes. So this is been widely seen in biological system, two popular things which makes our life possible on Earth. So that's why the topic is it's all about proton concentration gradient. Without this concentration gradient, we won't be able to live. Okay, so one thing is the production of ATP, I'll come to that in a minute, depends on the mitochondrial biochemistry. Mitochondria is a bimembrane, lipid bimembrane organelle. In that one, in between the two membranes, the proton concentration gradient is constantly made. So they are trying to conserve the energy, and that happens through a biochemical reaction which is happening inside mitochondria that pumps the proton into the intermembrane space. So now it is building the energy, and later on we will see this energy will be used to produce ATP. ATP is the currency of energy. I am speaking here, you are walking or listening, everything needs ATP. You can ask my biochemistry students how important ATP is. So similarly, you can see that when the same process happens in chloroplast as well, where the photosynthesis originally capturing the solar energy and conserving that in the form of proton gradient. So you can see that proton being pumped into the lumen of the chloroplast, so the proton is saving energy in that form. So both processes leading to that one. So here we captured the energy which is naturally happening in the biological system, as most of you know. One other process happening in our environment is in some bacteria. Some bacteria carry proteins called rhodopsin, and this rhodopsin belongs to a kind of big class of protein called opsins, and these opsin proteins are found even in your eye, your retinal cells. In retina you have opsin protein. That opsin protein changes its conformation when the light strikes them. Okay, the pigment which causing this conformation is called the retinal, and that goes from all trans conformation to all cis conformation over here. And this is how they make the change in the protein structure, and that is what the neurological signal going to your brain, and you are seeing vision. Okay, so that is the science behind that. And this protein is found in some bacteria for different purpose. They use them to harvest solar energy and make it chemical energy in the proton concentration gradient, so that they can use to produce ATP. So that is found in, say for example, this is one of the type of bacterium, Halobacterium salinarum, and it produces this bacterial rhodopsin protein, which is kind of a purplish color. And you can see that a well-populated lake, which is pinkish in color because of this Halobacter salinarum is growing very densely in that one. So this organism uses this bacterial rhodopsin. Using sunlight, they pump the proton, saving the energy. So as I said, there are proton gradient generators and some proton gradient users. So once you save the energy, you're going to use that energy to generate a commonly known energy form, that is ATP. And this is synthesized by ATPase. Once again, this ATPase is found in mitochondria. It is very much like the molecular motor of the bacterial flagellum I talked earlier. Okay, so again, when the proton flows, the motor moves and the ATP is synthesized, again, as we saw earlier. So how is this wonderful thing happening? You know, you have a bunch of proteins and some proton, hydrogen ion flowing through, and ATP is synthesized or energy is conserved. What is happening molecular-wise? So there's a video clip, I hope not to take too long time, just we will look at a segment of this one.
Many species of bacteria propel themselves through their environment,
shorter time here,
which rotates. The motor derives its power from a proton gradient across the membrane. In this example, a high concentration of protons exists outside, and a low concentration exists inside the cell. The protons flow through the interface between two types of proteins, called mote A and mote B, that make up the stator. Mutational studies suggest that a conserved aspartic acid in mote B functions in proton conductance. Each stator contains two mote B proteins, and therefore also contains two of these important aspartic acids. Although the molecular mechanism of rotation is not known, one possible model describes protons moving through the channels in the stators and binding to the aspartic acid in the moat B proteins. This binding causes a conformational change in moat A proteins, resulting in the first power stroke that moves the rotor incrementally. At the end of the first power stroke, the two protons are released into the cytoplasm. The proton loss causes a second conformational change that drives the second power stroke, once again engaging the rotor. Although the mechanism for motor function is not yet certain, many details of...
So we saw the mechanism how it happens, so that when the proton flows, the protein changes its conformation. So it's actually most of the biological function depends on protein conformational change. So for non-biochemists, protein conformational change is simply, you have a protein like this, something happens to the protein, so it kind of opens up, or like this, goes into this one. So it is kind of a motion, and that is going to do a lot of things like you're making my movement, speaking, or sensing various things. Everything depends on protein conformational change. So that's important. So this is a naturally known phenomenon, though it says the molecular mechanism is still being investigated. So why we are interested here at Eastern, okay, so why we ended up, or what we wanted to achieve from this phenomenon. So this is all started with exploring a bacterium. We study this bacterium called Colobacter crescentus. It's a very beautiful, unique organism. And it has a lot of features, two interesting features you may be interested. It produces one of the toughest known glue on Earth. And this glue can withstand 5 tons force on a square inch. Technology people need to jump on it and find out, okay, how it works on that. So it's a very interesting protein it produces to hold itself. And also, it doesn't need a fancy hamburger or anything to live. Okay, you can feed them like toxic organic materials, like which can cause cancer on us. But you throw this one, it will feed on it and live happily with that. Okay, so it really breaks that into useful organic compounds. So it's a very interesting organism to look into that. So we study the use of complex carbohydrate like cellulose, xylene, which is coming from plant biomass. So we study that aspect, how this organism uses this carbohydrate source. And based on that one, we came up with this model. This model may look a little complicated, so let me make it a little simpler here. So what our data suggests is this organism takes up gluco-oligosaccharide, that means a long chain of carbohydrate into the cell, into the periplasmic space, and where it breaks that using enzyme into simple sugar, glucose. Like you take starch every day morning when you take your toast, you are taking starch and you are breaking into glucose. Very same thing happening, but it is breaking plant cellulose, which you can't even digest. Okay, so that is happening here. And the glucose is converting into, glucose is used for the energy production. So this is how we entered into this particular research. So this is a result from a different study we learned. And if you look at this one over here, I mentioned this organism takes the gluco-oligosaccharide into the periplasmic space using what is known as TONB dependent receptor. TONB dependent receptor, though it's fancy, it is a simple membrane transporter. So it transports material into the cell via that membrane transporter. So a little bit detail about membrane transport, this particular membrane transporter. So as a name says, TONB dependent transporter, so it has a transporter component and it depends on a complex known as TONB complex, another set of protein inside. And this TONB complex is somewhat a molecular model like the flagella assembly, but it is not that fancy as a flagella assembly, and it doesn't do much work. What it does is when the proton flows through this machine, it undergoes some conformational change, and this conformational change is extended through this arm to pull a plug at the end of this transporter to release the substrate. So the substrate will come in and sit there, and it can't get into the cell until this TONB goes and opens the door for the substrate to get into the cell. So that is how this is working. So we found out this Colobacter crescentus using this TONB dependent receptor to import material. So we wanted to couple these two kind of techniques, like a TONB dependent receptor as well as the bacterial rhodopsin type of solar energy harvesting. So we put forward this hypothesis that proton gradients generated inside synthetic membrane vesicles by solar energy, using solar energy, can activate PMF, proton motive force. That is what, when the proton flows through the protein, it causes the change, the conformational change. Can use that one to make the protein to move. So to show that pictorially, this verbal hypothesis, that you can see that you have an outer membrane, in the inner membrane you have this proton pumping protein like bacterial rhodopsin, which can generate a proton concentration gradient, and that will be used by the TONB to activate this TONB dependent receptor. So this is our hypothesis we are putting forward. So as we know, hypothesis anybody can put forward. The thing is that testing and finding whether it's true or false is the biggest thing. So as Enrico Fermi said, sometimes it only you need to put forward the hypothesis. Whether you prove it or not prove it, you're going to make a discovery anyway. Okay, so it's a fact, it's going to come out of that discovery. Okay, so that is one thing to take up. Or sometimes I hear even my research students mumble this one in the lab. Okay, so I wonder, okay, this is maybe that's true too. Okay, so so we wanted to go forward with this one. So even though this, is it possible? Can you couple this one? Can you make the proton to work this one? There are a lot of work being done in this area. Not a lot, at least to some extent. Like some research groups have shown this TONB system in lipid membranes, and they make it somewhat sense the signals and so on. So there are some research, you can see that pretty recently someone showed that in 2013, and you can use this energy dependent motion of TONB. So there is progress in this area. So we are not completely proposing some outrageous proposal here. And technologically, we are all talking about membrane proteins. Dealing with membrane protein, it's a pain in biochemistry. It's very hard to make the protein and work with the protein. So one of the important technique in handling this protein is called nanodisc technology. So nanodisc is you can, instead of having a vast area of membrane structure, you can have a very limited membrane space. In that one you can embed, you can put your protein in. So you don't need to work with a big membrane structure rather than a smaller confined area. So here it's showing that how the membrane is confined with a protein, and another protein you can insert a membrane protein into this one. Again, this technique is pioneered by, not too far from us, University of Illinois, Professor Stephen Slicker's lab, and they really pioneered this technology, and they put various membrane protein into this membrane nanodisc, and they characterize, and they are even applying for many different things. And so that you can see that even bacterial rhodopsin, the protein I was talking about, and also proteorhodopsin have been used to synthesize this nanodisc. So it is, again, a one step forward for us to put together this whole idea. So how we wanted to do this research? We are planning to approach this one in a stepwise process. First, we have to get the proteins necessary, and we have to put them together in a membrane nanodisc, and then we need to make them work. So it is, again, a long-term process. We need to get the protein and work on it. So we are progressing on this one, and finally making them communicate between these two TONB system and the bacterial rhodopsin proton gradient is a challenging step. So what do we wanted to do with this one if we ever succeed in this one? So again, if you look at our model what we talked earlier, you can take the gluco-oligosaccharide into the intermembrane space, and you are breaking glucose. we can apply the same principle to generate renewable fuel. For example, you take cellulose and you put this vesicle containing container with this cellulose, you put the vesicle, and the vesicle will take the cellulose inside and break it into glucose, because you pack that with enzyme, this BGL enzyme that breaks glucooligosaccharide into glucose. So you're generating glucose or xylose, which can be used for renewable energy production. And again, you have noticed that one, the energy input for this one is 0, or it is actually completely solar energy. You are not putting any chemical thing, or you are not powering into a plug or anything like that. All comes from solar. So that is the overall idea. Or else a crazy idea, you can see that you can put together flagellum into the vesicle and generate something like an artificial bacteria. Okay, so you when you power the solar power, you can see this bacteria just running around, maybe in future. Okay, so long, it is a long kind of a too fancy hypothesis, but it is someday it's possible, but we have to take the first step forward to go ahead. Okay, so that's about how our research and how we wanted to bring that together. And so what are the current progress in this one? We are in the process of getting these proteins incorporated into nanodisk, and Susan's just left and she's working on that along with several other students. And we have already had discussion with Dr. Das. He's a postdoctoral fellow formerly at Stephen Sliger's lab at UOY. Now she's a faculty at comparative biology at the Veterinary School of Medicine in UOY. So she recently visited our department. She's enthusiastic to collaborate with us. So that's a big jump for us. You know, a lot of technological things, we can just rely on that. And got some funding for this one from PIF grant, and we current grant is pending, so we are hoping to get that money so that we can move forward. Okay. So anyway, I end up with this one with a quote from Dr. Abdul Kalam, the president and the scientist of India, that he said that you've got to dream first before your dreams comes true. If there's no dream, when will it come true? So you've got to dream. So we are dreaming it. Hopefully it will come true someday. Thank you very much for your attention.
Thank you very much. Any comments or any questions, or I have tons of them. Before I say anything, would you allow me to use some of these quotes in my research and technology class?
The quotations are not mine, but it is
It's only public domain.
Yeah, that is public
Yeah,
domain.
very, very.
Yeah, none of them is mine.
Very good. And it's amazing how the more we know, how less we know. I mean, it's an endless thing. Any other questions, if you like? Very good. So if you have any other questions, you have our speaker's email and my email, and let's keep in touch.
Most of us.
Thank you.
Thank you.